Our next presentation is Aurion Biotech, and I'd like to welcome Greg Kunst, CEO, to the stage.
Thank you very much, and thank you for the organizers for inviting me to present today. Continuing the theme on transplantation, and we're going to talk about a different area, a different part of the body, and using cell therapy as a way to overcome the challenges of transplantation. These are my forward-looking statements. We're going to focus in an area around the eye, and this is called corneal endothelial disease. This is a very common form of blindness around the world. Today, it's addressed through corneal transplant, the most pervasive form of human transplantation. Unfortunately, in corneal endothelial disease with human corneal transplant, requires one donor for every diseased eye. And we only have, for every one donor available, there's 70 people around the world waiting for treatment. We're going to talk about a cell therapy that has been through the test of time. The original patients were treated 10 years ago. We are now into almost 6 studies, or over 6 studies now, with a number of patients that I'll walk you through that data. Additionally, we are approved in Japan. The technology came out of Kyoto Prefecture University in Kyoto, Japan, and we have approval. We're now reimbursed, and we start commercializing in Japan this past month. Additionally, we recently completed our phase 1/2 study. We'll report that data early next year. But really off the back of the data that we've created the last 10 years, we do have breakthrough status and RMAT status. We're one of the only companies in the world to have both breakthrough and RMAT, and we'll talk more about that in a second. We are backed by blue chip investors. More importantly, we think we have something that is very unique to cell therapy, and that's something that where we think we can scale massively in what we do. In our current iteration of our product, for every one donor, we can create over 1,000 treatments. This is an off-the-shelf allogenic cell therapy, and more so, we're doing this with a team of people who have deep experience in cell therapy and in ophthalmology. So why is the mass market nature of what we're doing so important? Well, the NIH estimates that 4% of the population over the age of 40 is prevalent for the most common form of corneal endothelial disease, and that's called Fuchs dystrophy. Fuchs dystrophy is a genetic disease. It affects both eyes. If we think about the underlying prevalence, that's about 11 million patients, or 32 million eyes in the US, Europe, and Japan that are prevalent for Fuchs dystrophy. On an annual incidence basis, we estimate about 500,000 patients, or a million eyes. Of those million eyes, about half of those would be in our total addressable market. But more importantly, we look to the right. We have 500,000 that we think we can address every single year on an incident basis, but only about 70,000 are treated with modern corneal transplant in the US, Europe, and Japan. Now, we think about the underlying disease. What we're addressing is arguably the most important part of the eye, which is the cornea. Without a clear cornea, we can't see as human beings. But more importantly, the endothelium is arguably the most important part of the cornea. It's a protective layer that ensures that the cornea receives the right amount of hydration. We want the cornea to be hydrated, but not so hydrated that it ends up causing cell death and eventual blindness. And as humans, the number of cells that we're born with at birth is the highest number of corneal endothelial cells that we'll have in our lives. And as we age and as we face disease or trauma in our lifetimes, we lose corneal endothelial cells. On an average person, we lose about 1% to 2% of our corneal endothelial cells on average year over year. And more so, if one goes under cataract surgery, we can lose up to 10 to 15% of our corneal endothelial cells. Now, most of us, that doesn't cause an issue. But in some people that are predisposed, losing enough of these cells can cause corneal endothelial disease. And more importantly, the cornea and endothelium do not naturally replicate. And so what we're doing with our cell therapy is we're actually replacing cells that have died and have caused corneal endothelial disease with natural new cells. Now, as the disease manifests, all these patients will eventually, who end up with underlying disease, will eventually progress. And they progress in a stepwise fashion. Once the endothelium stops the pumping process in the cornea, the cornea begins to get cloudy, And we can actually see that when you look at people's eyes very carefully. From there, the patient starts to have weird artifacts in their vision during nighttime driving or low light reading. But eventually, all patients will progress toward losing vision, and in some parts of the world can go completely blind. There's actually estimates between 8 and 12 million people in the world today are completely blind from corneal endothelial disease, primarily because of lack of access to, to donors or to treaters. Our cell therapy is restorative. Once we inject the patient with our treatment, the process is reversed. The endothelium start repumping. The cornea clears. The second-order aberrations are cleared. But more importantly, vision is restored. What we're doing, as we said earlier, though, is not only are we addressing an underlying problem around supply around the world. Obviously, there's not enough transplants around the world. And by the way, the transplant numbers haven't changed in 30 years, and there's really no prospects for the donation pool to increase around the world. But what we do is, obviously, we're able to scale massively. Again, with one deceased donor, we can create 1,000 treatments. We think this number will probably increase over time. Just with our current scale, we can address all of the disease patients around the world into perpetuity. But more importantly, what we also do is, because this is you'll see here in a second, is a straightforward procedure, it can be done by a number of ophthalmologists, frankly, most ophthalmologists around the world. Why that's important is here in the United States, there's only about 300 corneal specialists doing corneal transplant. Conversely, there's about 8,000 ophthalmologists do cataract surgery. We believe not only by expanding the supply of cells, but also expanding the number of treaters that are available, we can have a demonstrable impact on patients that should be treated that are not receiving treatment today. And more so, this is a minimally invasive approach. This is not a transplant. This is an injection of cells that allow us to move this much earlier in the treatment algorithm. Now, here's the procedure. It's 3 straightforward steps. In step 1, we create a little incision in the eye. This is a self-sealing incision. That incision, we put a little device that allows us to polish off the diseased endothelium. We're trying to remove the old ineffective endothelium, and then we inject cells. The cells very quickly migrate straight to the stroma and form a monolayer. And to facilitate that adhesion, we put patients face down for up to 3 hours. We think in the real world this will be less, maybe as decreased to 1 hour, but needless to say, this is much more patient-friendly than modern corneal transplant. Modern corneal transplant, patients have to lay on their back at least 3 days after treatment. Now, how we actually create the cells, again, we take a deceased donor, very similar to what's done in corneal transplant. Once we take the deceased donor, we remove the endothelial cells off the cornea, and through a proprietary mechanism, we're able to turn on the replicative process, but more importantly, turn it off once we're done. We go through multiple passages. It's not just as simple as removing contact inhibition. There's a series of scientific problems that we have to overcome, and this is really the genius of what our inventor was able to discover, and really where our IP resides. Additionally, again, we can do this at high scale. We aliquot these into single doses and then send these all around the world. And we can do this using the modern logistics systems that allow today corneal transplants to be sent to every part of the world. Our data is if I haven't convinced you that we're working on something that's truly transformational, I think as we go through the data, my goal here is to convince you of that. We've treated over 130 patients outside the U.S. and Japan and El Salvador. We've also treated another 97 in our phase 1/2, and that in the United States and Canada. That data is baking as we speak. This is the first 11 patients. This was published at 2 years in the New England Journal of Medicine and 5 years in Ophthalmology, our highest tier journal in our field. What we're looking at is really two features, clinical feature endpoints that demonstrate the effectiveness of what we do. And so we're looking at both central corneal thickness, that's an anatomic function, and then visual acuity, which obviously is the goal that we have. We want to restore vision. And so if we look at corneal thickness, what we look for in corneal thickness is a cornea somewhere between 500 and 600 microns. These corneas in the first 11 were 743 microns. That's quite endeminous. These are people that are clearly sick. By 6 months, they're back to healthy ranges, and they stayed that way through 5 years. If we look at visual acuity, this is where it becomes most meaningful. These patients were 2,220. 2,220 is legal blindness. By 6 months, they're 2033 and stay 2030 through 5 years. These patients now are out to 10 years. We'll report this data later on this year, but I can tell you at 10 years, they continue to do equally well. Again, we don't tend to see this as often in corneal transplant. Patients tend to start to need revision surgery by year 5 in modern corneal transplant. Additionally, if you look at all the studies that we've run outside the US, the Japan study and 2 in El Salvador, what we're trying to look at is similarity of results. This is looking at central corneal thickness across the 3 studies. You can see an immediate restitution of normal central corneal thickness by around 1 month and stays that way through 12 months. I can tell you as well, as we look at the El Salvador patients, again, those patients continue beyond 12 months to have similar results. This is the most important slide in the entire presentation. The approval endpoint for anything involving vision and therapeutics and ophthalmology is the percentage of patients at 3 lines of improvement. 3 lines of improvement is considered clinically relevant and can't be masked by retest error. And so in this particular analysis, what we see here is by month 1, about 50% of patients have a 3-line improvement in vision. But more importantly, by month 12, we look at 80 to 90% improvements in 3 lines. That is unheard of in the field of ophthalmology. Our greatest drug that we have in ophthalmology is arguably the anti-VEGFs used in macular degeneration. If we look in that particular category, which happens to be a $10 billion market, that particular category, you go back and look at the original pivotal trials, they were seeing 30 to 35% of patients at 3 lines of improvement. To find a therapeutic that can deliver 80 to 90% is something that we only see in surgery in ophthalmology. So remarkable improvement in vision. And again, this continues trial by trial. So where are we at? We came off last year finishing our randomized control trial in El Salvador, dose-ranging phase 1 trial. Again, we recently completed our phase 1/2 in the US and Canada, 97-patient, 5-arm, fully masked randomized control trial. We'll report that data later this year, early next year. Additionally, what we've done is we've recently got breakthrough in RMAT. I can tell you one of the questions people ask is what's the difference between breakthrough and RMAT? While the benefits are similar between breakthrough and RMAT, we certainly believe the bar to get breakthrough is much higher and really requisite around our pathway to approvability. Interestingly enough, you look at all breakthroughs, 90% of breakthroughs drugs end up getting approved. We just came off the back of launching in Japan. We've done initial commercial patients, and we'll continue to expand our scale in Japan. And we just recently, actually this past week, came out of a cryo study in El Salvador looking at a cryo version of our program that we plan to launch post-approval. We are backed by esteemed list of investors led by Deerfield and Alcon. We think this company continues to really move in the direction of being able to be transformational in an area of medicine with high unmet medical need, and this is coming soon to countries around the world. With that, I thank you for your time and attention. Have a great day.